just for my sanity, yep. All right, good. Just leave that here. All right. Does everybody have a cheat sheet that needs a cheat sheet? Would any of the peanut gallery like a cheat sheet? Would you like, here, pass them down, pass them down. Yes. It's on my Twitter, Darshu. It's on the link is on my Twitter, so you can watch it live or later. Lovely. Good morning. All right, so all of the committee, with the exception of Adam, are here. Adam's going to be late. She told me to start with Adam.
We're ready to start. Okay, all right, yeah.
Yeah. He was a half hour late to my proposal, too.
Is your middle name Shannon?
You bet. It's Joan. It's my mother's name. Where's Joan? Dead. This is her sister, though, in the black. That's my godmother. She's the mother steppin'. Do it. God help him. What's he gonna do? Gotta get ready. Go get some good.
Good morning, everyone.
It's fine.
Good
Well,
morning. Can you hear me?
Longer ago it was. Yep.
Good morning. So I'm Spencer Nyholm, and it is my extreme pleasure to introduce Sarah McAnulty today to defend her dissertation, A Tale of Two Symbioses, Development and Maintenance of Bacterial Partnerships with the Hawaiian Bobtail Squid. So Sarah joined our program after receiving her BA in Marine Science with distinction and a minor in biology from Boston University. She spent time in Berlin, Germany, working as a technician in a lab before she started in our program. And the first thing that was very obvious to me after meeting Sarah for the first time, that she's very passionate about science. And anyone that knows her well knows that she loves nature and she loves animals. And there are a bunch of slides that I'll show you that are evidence of that. So here Sarah is with a whale. Here she is loving a crab with an owl on her head. Even animals that are alive, like this bear statue that's in my front yard, or scary animals, she loves them all. But we all know that there's a group of organisms that she really loves the best, and those are the cephalopods, of course, and the squids. She just has so much passion and enthusiasm for them. Other animals just don't quite measure up to squids and cephalopods. And she loves communicating the science of cephalopods to the public. She even has a squid tattoo on her arm of her study organism, the Hawaiian bobtail squid. That's dedication. Anyone who has seen the Squidmobile knows that this is one effective mechanism that Sarah has used to communicate her passion about science and squids and her research. She's been sought after as a speaker about communicating science and also cephalopods. So she's been interviewed on Science Friday twice. And here she is at a live event in New York for Cephalopod Week that's held every year for Science Friday. She even wrote a children's coloring book on cephalopods that's published. But, and she's been, Science Magazine asked her to write a book review about a book that was published, Squid Empire. So she reviewed that book for Science. And she's been interviewed on a number of podcasts about her research and her love of science. Now, in 2017, Sarah took this passion to the next level and founded this organization, Skype a Scientist, which many of you are familiar with, that its goal is to basically pair scientists with classrooms around the world. And in 2 years, a little over 2 years, it's taken off amazingly. So it's now in thousands of classrooms around the world involving thousands of scientists. And this is, I think, an amazing feat in such a short time. She's been interviewed about this. She has won accolades. She's a sought-after speaker now for science communication and Skype a Scientist. Here she is at an event, I believe, promoting women in science. She's been written up in Nature. Here's a Nature Career Q&A, Squid Biologists Connecting Schools and Scientists Worldwide. And this past year, so this year actually, she was the recipient of the NEB, New England Biolab's Passion in Science Award, which is an award that's given out to folks from all over the world. And she received this for science mentorship and advocacy in 2019. So Sarah has done a phenomenal job in communicating her science and in science communication. But today we're here to talk about her scholarship. And this is a photo that was taken this summer at the Marine Biological Laboratory in Woods Hole with some members of my group. Sarah's right there. And you see the squid in the background. And the MBL is a pretty special place. And while we were there this summer, I asked my students to indulge me, and we went up to the library. And on the floor of the MBL library, there's this case that has this photo. That gentleman in the background is Thomas Hunt Morgan, and he won the Nobel Prize in Physiology and Medicine in 1933 for his discovery that basically genetic information is transmitted through chromosomes, and for the development of Drosophila as a model organism. But Morgan is in my lab's direct academic lineage, so he's Sarah's academic great-great-great-great-great-grandfather. And he was an amazing person. And the reason I bring him up is many people know him for his work about Drosophila. But what people may not be familiar with is that he studied a number of other organisms. He was very passionate about studying a variety of different animals. And he spent 55 summers at the MBL. And he would actually, every summer, he believed also in experimentation. So he was one of the first invertebrate zoologists to apply experimentation to studying these questions. And he believed in long-term experiments. So he would actually pack up his fruit flies in sugar barrels and ship them to the MBL each summer so that the experiments could continue and they wouldn't have to stop. His students and he would take a boat to the MBL, and they would take with them from New York cages of mice and pigeons and rats and any other animals that he was working on. And he spent the summer there continuing these experiments, but he was really passionate about marine invertebrates, and there's a very rich literature from him. And he on marine invertebrates, and he realized the importance of working on model organisms. And so he sought out organisms that allowed him to answer the questions that he wanted to answer. And I bring that up because Sarah also got her start at the MBL. So she was an intern there for 2 summers, and she worked in the lab of Roger Hanlon. And as an undergraduate researcher, actually is a co-author on 2 papers from Roger's lab from that work. And when Sarah joined my lab, she wanted to ask questions about how symbiosis in the Hawaiian bobtail squid, how these interactions, how they develop through time. And up until that point in 2013, when Sarah joined my lab, we were looking usually at colonization of the Hawaiian bobtail squid by these bacteria in the short term. And Sarah took up the challenge of raising the bobtail squid in our lab through multiple generations, which opened up a whole new toolbox for us. There are very few people in the world, actually, who have been successful in raising not only the bobtail squid, let alone any cephalopod, through its entire life cycle. And Sarah is one of the select few who have done that. And so her dedication to the animal husbandry in the lab really allowed her to ask some very significant questions about these two different symbioses that we're studying and how they develop in the long term. And we're going to hear a lot about that work today. So Sarah took that information. She went back in the lab. She's a great microscopist, so she did a lot of work with working on blood cells from the squid using confocal microscopy. She loves working with the animals, so these are actually the little tubes that we keep our baby squid in. So here she is monitoring the squid. There's a lot of teamwork involved, so she worked very closely with Andrea Surya and Allison Kerwin in my lab on a lot of this work. And she has presented her work at a number of meetings. So she's presented her work at 14 presentations that I counted at regional, national, and international meetings. For many of these, she's won Best Poster or Talk Awards. And so here we are at a conference in Hawaii in 2016. Here, she and Allison were both recipients of a Best Poster Award at a Gordon Research Conference on Animal Microbe Symbioses in 2017. Here's another conference in La Jolla last year, commemorating the 30th anniversary of the Squid Vibrio system. And Sarah was not satisfied just working on the Hawaiian bobtail squid. She came to me and she said, I want us to look at the symbiosis in other cephalopods too. And I had done a little bit of work in Japan, and she said, I want to go to Japan and collect it. I said, well, I don't have funds to do that right now, but I tell you what, if you can raise the money to go to Japan, then we'll go. I didn't think we were going to Japan, but we ended up going to Japan. Because of course, when Sarah puts her mind to it, she gets things done. And so she raised the funds to go to Japan. So here we are in Tokyo collecting squid and actually eating some squid at the same time. And in Okinawa, with our collaborators from the Okinawa Institute for Science and Technology, enjoying a nice dinner after being out squid collecting. So Sarah's work has so far contributed to 4 publications in my lab, with 3 more either submitted or in preparation right now. And she's been a tremendous resource in the lab. She's been a fantastic student. I'm very proud of the work that she's done, and I'm very much looking forward to hearing her talk today about the research that she's done here at UConn. Thanks, Sarah.
Okay. Okay, all right. Well, thank you so much for the introduction. That has made me slightly emotional, but that was very nice. All right, so are we all ready to hear about some squid today? All right, excellent. So today I'm going to be telling you about 2 symbioses in the Hawaiian bobtail squid. So who here has ever taken a probiotic or has heard of their microbiome? A pretty solid number of us, right? So we know that bacteria are very important for our health. They help us with digestion. They help protect us from, oh, I've got one on, don't worry. Anyway, and they help us with protecting us from pathogens. And we know that altered communities in our bodies of bacteria are associated with a number of diseases, including obesity, diabetes, MS, autism, and the list goes on and on. So what I've tried to understand during my time at UConn is how animals communicate with their bacteria. And the animal that I've used to understand these associations is the Hawaiian bobtail squid. This squid may seem very different from us, but they're actually a really powerful model for understanding this type of communication because they have not 1, but 2 major symbioses in the same animal. The first symbiosis is with just one type of bacteria. It's a bioluminescent species called Vibrio fischeri. And the second is a consortium of bacteria that's only present in the female squid. And we'll talk about that more in a moment. They're a great model because we can raise the squid either with the bacteria or completely without the bacteria, which allows for us to kind of play with what happens and how bacteria influence the development of a squid. And we can also manipulate the genetics of the bacteria so we can see what really is important mechanistically for this communication. So, like I said before, there are two major organs that are important for symbiosis. The first is the ANG, the accessory nidamental gland. I'll just be calling it the ANG for the rest of this talk. And also the light organ, which is where the bioluminescent bacteria live. The light organ is two-sided, has two lobes, and both of these lobes have three crypts, which are basically like little pockets where the bacteria live throughout the squid's life. There's just one species of bacteria there, and they glow all night. And so if the squid has the bacteria present, it's able to camouflage with the moonlight coming down from above. So a predator swimming below, instead of seeing a little squid-shaped silhouette, doesn't see anything at all. The silhouette is obscured. The second symbiosis, only present in female squid, the ANG, is a consortium of bacteria. And so within the ANG, there are a number of tubules, and each tubule is home to a different type of bacteria. And there's a number of things that these bacteria do, but one of the main things that we've been studying is the production of antimicrobials. So each sort of bacteria is responsible for making perhaps a different type of antimicrobial that then protects the baby squid as it develops. So when the female squid lays her eggs, she will wrap the baby squid in a series of concentric circles of jelly and then add bacteria to that jelly. And then they produce the antibiotics and protect the baby squid. So today I'm going to be telling you two stories of what I've worked on during my time here at UConn. The first, all about the ANG, how it develops, and how bacteria influence that development. And the second will be all about the immune system of the squid and how immune cells are able to identify beneficial bacteria and others. So let's first go into a little bit more detail about the ANG. So the ANG, again, this organ here, you're going to be seeing a lot of pictures of ANGs today, so you might as well start to really internalize that picture right now. It has, like I said, a consortium, and it's acquired from the environment. So when the squid hatch, they develop a little bit, and then they acquire that bacteria from the environment. They're not born with it. And so we want to understand how that happens. So here's a zoomed-in picture of the surface of the ANG. You can see here there's like a white type of bacteria here, and there's more tubules over here that are more orangish peach-colored, and they produce different pigments that allow us to see the different colors that are being produced there. And so if we take a cross-section and kind of look down the tube of the ANG tubule, you can see that it's full of bacteria. And if we do what's called FISH, which effectively allows us to, not like the animal, like a fluorescence in situ hybridization FISH, it allows us to look at different species of bacteria all at the same time. And so here we can see this blue is one type of bacteria called Verrucomicrobia, and the red is a different type of bacteria, the alpha proteobacteria. So we can see that the bacteria partition into different tubules. They don't all mix in together. So there are four major groups of bacteria that are present in this organ: the alpha proteobacteria, the gamma proteobacteria, the Verrucomicrobia, and the flavobacteria. And here you can see, again, a diagram of the egg, and then all of these little dots are bacteria that are suspended in those jelly coats. And so this is each a layer of the jelly coat there. Okay, so during my time, I've been characterizing the development of this organ. I've raised a couple hundred squid during my time here, and then I've been looking at their development as they grow. The main things I wanted to understand were how this organ develops, how bacteria are recruited, and how the bacteria influence the development of this organ. So before we really get into the nitty-gritty, I'd like to go over a brief lesson on squid anatomy. The first, here we have our Hawaiian bobtail squid. And what I'll be referring to is the mantle length of the squid. The mantle of the squid is where all the organs are located, and it's the measurement from behind the eyes to the back of the body. This is basically a readout of maturity because they get bigger as they get older. They max out around 30, but we consider maturity 20 millimeters mantle length. So when you see ml throughout the talk, that's all about the size of the squid. The other really important thing for the biology of this animal is that their mantle is open to the seawater all the time. Because when squid breathe in, when you and I breathe in, we breathe into our lungs, which is kind of all, partitioned. But when a squid breathes in, it's like air is entering, or water is entering their whole torso, effectively. And so that allows all of their organs to interact with the seawater, and therefore all the bacteria that live in the seawater all the time. So as I was raising these squid, I was sacrificing them as they got older. And the first notice of an accessory nidamental gland, an ANG, was this small nub of tissue right here, this little bud. And they were full of furrows. And so these were only a couple hundred microns. You couldn't really see them with your eye if you looked at the squid. You had to use microscopy. And throughout this talk, there's going to be cartoons on the side here of what we think is going on cellularly. And then as the squid got a little bigger, we first started to see the signs of small pores forming right in the center of these pads. So the bud before was kind of sticking up like this, and then at this stage, they were starting to flatten down, and you would see two of these circular pads. And if we zoom it in a little bit, you guys can see the circles a little better. So that's where we saw those pores. As the squid gets larger, those pores multiply until there are about 1,000 to 1,500 pores per animal at what we consider like the maximum recruitment phase. If you take a cross-section of animals at this size, you can see these pores are full of cilia. And so we know that cilia are very important for other parts of the squid symbiosis. So for the light organ symbiosis, cilia are very important in recruitment. And so we think that probably in this symbiosis they're important too, but we haven't characterized how that's happening yet. And so we think during this phase, when there are so many pores present, that this may be when the squid is recruiting bacteria from their environment. At a larger sizes of squid, around 12 to 15 millimeters mantle length, we're starting to approach maturity. You start to see these two layers in the tissue. This top layer here is the recruitment layer, so that's where all of those pores were in the cilia, and then beneath that, you have developing tubules. So if you take an EM of that, you have the tubules here full, or the pores here full of cilia, and then down below you start to get these colonized tubules where bacteria is replicating. Even here in this blow up, you can see right here, this little peanut looking guy is a bacterium that's splitting. So they're really dividing at this phase. So pay attention here to the pink line. Above that is our recruiting area, and below that is the developing tubule area. When a squid is sexually mature, that recruiting tissue is no longer present. So everything that was above the pink line in the last slide is no longer there. And so we think that once a squid is fully colonized, they no longer need to recruit bacteria from the seawater because they already have all the bacteria that they need. So we also wanted to know what happens when you raise a squid in the absence of the bacteria that they would encounter in the wild. We wanted to know if they would still develop this recruitment tissue or if it wouldn't form at all. So I raised squid in the absence of their natural bacteria, and we still found that this recruitment pad still forms. One neat thing that we noticed during this experiment is that, can you see in this picture here, the area between the pores is pretty smooth. And then here, it starts to become a little disorganized. It looks like it's kind of starting to fall apart. And so when we look in higher resolution, this is another cross-section, you can see that instead of having those smooth pores that reach the surface, the epithelial cells are beginning to become apart. And so we think that what we're catching is the loss of that recruitment tissue. And so by the time a squid reaches adulthood, in the absence of bacteria, there's no recruiting tissue or ANG at all. So So here's our model for understanding how these squid are developing ANGs. We think they start around 2 to 3 weeks at 4 millimeters. Around 5 to 6 millimeters, we think they have the first sign of pores, which then become much more numerous. We then have this 2-layer phase. And then we have a fully formed ANG, which then the bacteria will continue to develop and become more pigmented over time. So the second part of this project, I wanted to know how bacteria influence this development. So I raised squid in a number of controlled conditions with various different bacteria. And so what I did first is I raised squid on autoclaved sand. Autoclaved just means that I sterilized it with heat. So I killed all the bacteria that were there. And then I raised the squid on filtered seawater. So all of the bacteria got filtered out. And then I sacrificed the squid at sexual maturity and then looked to see what their organs looked like inside. And what we found was that the squid were completely lacking an ANG. So this is kind of tough to see, but this is what a squid looks like when it has an ANG, and this is what a squid looks like when it doesn't. You can kind of see the tubes of the digestive gland beneath, and you can see it even better in this next slide. Can you see how they're right here? We have these tubes. That's the digestive gland that lays beneath where the ANG would normally be. And so the next thing I did, I went back to Hawaii, and I collected sand up from a beach on this area of Oahu right here. And so after picking out all the cigarette butts that were left on the beach, I raised the squid on this sand. We did this because the really fine sand is really easy to raise squid on because the rocks in the sand aren't as big as them, and then you don't end up with a squid that gets squished by its own sand. So it's really nice for husbandry. But it turned out to not develop any ANGs because the sun-baked sand, we don't think had any living bacteria present, or any living bacteria that were relevant to our squid. So we went back to Hawaii again, twist my arm. So we had to go and collect sand from beneath the low tide line because we wanted sand that had the same bacterial community present that you would have in the ocean. My lab mate Allison had previously shown that the bacterial community that's present in the sand has a lot of overlap with the bacterial community present in the ANG. So we thought that the place that the squid were getting their bacteria would be from the sand. And so when I brought this sand back, raised the squid on it, sure enough, we got ANGs to grow. But we wanted a little more information other than just success. We got them to grow. So Allison sequenced these animals, and we're looking here at what's called an NMDS plot. So it's looking at the similarity between bacterial communities, and then it groups the samples based on similarity. So here we have wild squid, so squid that were caught from Hawaii in blue. And we have the squid that I'd raised in the lab on the Hawaiian sand in pink. Don't worry about the gray bars. Those are from different things that aren't relevant to today. So as you can see, of these groups, the pink is grouping mostly with the wild caught squid. So that shows that we can do a pretty decent job of recapitulating that bacterial community in the lab from the Oahu wet sand. So once we were able to develop an ANG in the lab, I really wanted to see if we could add in a specific member of the community, so to see if we could, further down the line, really start playing with some experimental manipulations with strains of interest. So we took a strain that my lab mate Andrea Surya had isolated from squid jellycoats called JC1. We liked this strain because it was this bright blue-black sort of color, and that's pretty different from all the other strains that we isolate from this community. So it was pretty much just convenient for experimentation. So I added 10,000 cells per milliliter every single day to these squid from the time they were about a week old up through 20 millimeters mantle length. And then I took the organs out of the adult squid, homogenized them, plated them, and what we found was that in squid that I added the bacterial, this bacterium to, ended up having anywhere between like 30 and 50% blue colonies on the plate, which is great because that means we can really enrich these communities with strains of interest. And just as a comparison, a wild squid would typically have like 1% blue colonies. So we really enriched these squid. So the next thing we wanted to do was see if we could effectively make a gnotobiotic squid, a squid that only has a couple species that we're familiar with. So I started with the alpha proteobacteria because these are the most abundant bacteria in the community, and no ANG developed. So I thought, okay, maybe they need a more diversified community. So I picked one of the alpha proteobacteria, one gamma and one flavo. For anyone who's been paying excellent attention, you might notice that we are lacking the verruco microbia. That's because we can't culture it. So we were unable to add that to the mix, but no ANG developed. And we also wanted to take a look at the influence of Vibrio fischeri, that's the light organ symbiont, on the development of this organ. So we raised some squid with Vibrio fischeri, some without. And the presence or absence of Vibrio fischeri had no effect on whether or not the ANG developed. Oh, and an important note here is that every morning at dawn, the light organ will eject about 95% of their light organ bacteria. So the mantle cavity is once a day full of Vibrio fischeri. So those pads that we saw earlier will always have Vibrio fischeri in the neighborhood. And so I wanted to see if that would affect it, but it doesn't. All right, so more sequencing. So we gathered squid from the wild and sequenced their ANGs to see if the bacterial community changes during development. So everything to the left of these red bars is a squid smaller than 20 millimeters mantle length, and everything to the right is 20 or more. So what you're seeing here, the smallest we would have had would be about like the size of a pinky fingernail, and the adults would be like a small lime. And so what we found was that smaller squid had relatively more verruco microbia, which decreased as the squid got larger. And as the squid got larger, more alpha proteobacteria were present here. Now this is all relative abundance. So this is a percentage at any given point. So over time, likely there was a lot more bacteria present overall, but it was just a relatively more verruco microbia at the beginning. So we were thinking that maybe verruco microbia is important for early colonization. So then we looked at the sand that we had used in experiments. We wanted to see what was present in sand that ANGs developed in compared to sand that ANGs did not develop in. And the main difference between these two sands was that in the no ANG development sand, there was no verruco microbia. And in the wet collected sand that did have ANGs develop on that sand, it had verruco microbia. So this is two kind of pieces of evidence that make us think that perhaps verruco microbia is the key to early development. They may be an early colonizer of the ANG that sort of paves the way for other bacteria to get in. We also look at how this development of this organ is similar or different to the light organ development. We've been studying the light organ for about 30 years. We know a fair amount about how this organ develops, how it all works. And so in the light organ, this is what it looks like when it first hatches, when that squid is trying to recruit bacteria from the seawater. And it has lots of cilia around these pores. And once bacteria enter the crypt spaces, they get a signal from the bacteria that induces a developmental program. And these appendages here are lost. The ciliated epithelium is also lost, so that you go from having, this is an actual picture of using an electron microscope. You have these appendages here, and then it's smooth by the time the animal has fully developed. And we know from what I've found in the ANG that when squid are young, they have this recruitment layer full of cilia, and then once the squid is an adult, the surface is smooth. So we have that in common, but we don't know how the signaling happens in the ANG. We'd also like to look more at how the cilia are important in this symbiosis. So this is a little pet project of mine that Spencer had mentioned in my introduction, all about looking at more cephalopods than just the Hawaiian bobtail squid. And this may have been slightly influenced by the fact that I wanted to go around the world collecting cephalopods, but we don't tell our funders that part. So I looked at the Hawaiian bobtail squid. This is the ANG of the Hawaiian bobtail squid. My lab mate Nidhi collected Uroteuthis from India. We collected the market squid that if you've had calamari this summer, you've probably eaten in Massachusetts. And then we collected 3 species from Japan, the big fin, or the oval squid, depending on who you are, what you want to call it, the cuttlefish, and also a different bobtail squid that we collected while snorkeling. And what I want you to get out of this slide is all the different colors that you can see in these different organs. So here in our little sepiola, we've got a lot of orange. We have more hot pink and red here in the Indian squid, but we also have hot pink and red over here in the Massachusetts squid. So we also wanted to know at the bacterial level what's going on here. Visually, they're strikingly different, but what do they have going on at the 16S level? So my former lab mate, or my current lab mate, Nidhi, this was started by my former lab mate, Allison, and then Nidhi took it over recently, has been sequencing these organs and then comparing the communities. And this is another NMDS plot, and what I want you to get out of this is that all of the bobtail squid, so the squid that look like a Hawaiian bobtail squid in their body shape, are blue and sort of clumping together here. And then our market squid that are pretty much shaped like this are over here. And then this one and this one are kind of weirdos because they were raised in a lab, so they might not have their normal bacterial community. But this really isn't enough data to make any huge conclusions from. So Nidhi and I will hopefully be collecting more squid in different places of the world so that we can really figure out what it is that determines what your ANG community is. Is it who you're related to? Do you have the bacterial community similar to people close to, or animals close to you on the tree? Or is it more about where you live? And so the reason we wanted to go to Japan was because there's a lot of different cephalopods all living in the same space. So they're in the same water, but if their bacterial communities are different, but any of those squid are more similar to animals that they're related to, we can kind of figure out what's important for what informs the bacterial community. So we're going to wrap up part one here, all about ANGs. We know that the ANG is recruiting bacteria during a colonization window when those pores are present. We know that the ANG tubules do not develop in the absence of the proper environmental cue that we were able to get from the Hawaiian sand. When you collect sand from underwater in Hawaii, we're able to develop an ANG. And once you get the induction of ANG development, we can add strains from the community in that'll be fun for later experiments. So there's a lot that we want to keep studying with the ANG symbiosis. We've really only gotten started with understanding what's going on in this symbiosis, which makes the two first-year grad students in our lab have so much to play with, and I'm a little bit jealous. So the first thing they need to do that I'm decidedly not jealous of is culture Verruco microbia. It's been a real bear to try to do. We have a former lab mate, Andrew, in the front row who tried it first, and then right next to him is Andrea, who tried it next. And then we've got Nidhi, who's been trying, and Derek, who've been trying for the last year. God help them. So hopefully they'll be able to do that. The other thing, we want to look at these ciliary flow dynamics, understand whether they're important for attachment of the bacteria, whether they're important for creating vortices to get that bacteria in. And we also want to understand how this squid is able to culture many different types of bacteria in these different tubules. It's possible that they make different microenvironments in those tubules. Maybe they're giving them different food. Maybe they're giving them different pH environments. That'll be fun to play with. And I'd also like to continue filling in the cephalopods of the world map. There's a couple species that are kind of on my wish list for things I'd like to get. And so that'll be dependent on little bits of funding we can get so we can travel and scoop squid out of the water in crazy places. So now I've told you all about ANGs, and it's time to go into the immune system. McAnulty's, flip to the last page in your cheat sheet. This is about to get real. Okay. So your immune system, all of our immune systems, are finely tuned to avoid disaster. If they're any more active than they currently are, you end up with an autoimmune disease. And if they're any less active than they currently are, you're going to end up being susceptible to pathogens. And so we have a pretty decent understanding of how the immune system is able to tell the difference between you and not you. But we don't have as good an understanding of how the immune system determines bacteria that's good for you and bacteria that isn't. And so that's really at the heart of what I'm trying to understand with this project. So let's talk about squid immune systems. They have one type of immune cell that's called the hemocyte. These hemocytes will traffic throughout organs and tissues, doing what a typical immune cell does, make sure everything's good to go. They also will wound heal. They're sort of a more multi-purpose immune cell than the immune cells that are present in the vertebrate immune system. They go into the crypt spaces. So here's that light organ. Oh, geez. That light organ cartoon again. They enter these crypt spaces where they're seen to interact with bacteria, but they don't destroy the bacteria present. So they know to leave them alone. And what I want to understand is how these immune cells are able to know that that's a bacterium that you don't want to mess with. So previous work from my PI, Spencer, during his grad school time, looked at binding of hemocytes to bacteria. So here we've got Vibrio fischeri, so this one in your cheat sheet. This is our beneficial bacteria, and the hemocytes bind fewer of the beneficial bacteria than this other marine bacterium, Vibrio harvii. And then he had a really cool result with his co-authors. He found that when he treated the squid with antibiotics to remove the bacteria from the light organ, over the course of 5 days, the squid hemocytes no longer bound few of the symbiotic bacteria and more of the non-symbiotic bacteria. It did about the same to both. And so that indicates that the presence of Vibrio fischeri in the light organ continually influences the immune system to not attack Vibrio fischeri. So I thought that was a really cool result, and I wanted to get a little bit more information about that. So I developed these new assays that use live hemocyte microscopy to view this interaction. And so I take a squid, I take a blood sample non-lethally, the squid's fine. I then add a fluorescent dye so I can see the immune cells with the microscope. I then incubate the hemocytes with these various types of bacteria and then watch what they do for about a half hour using a microscope. And so here's what the data end up looking like. In pink, we have the hemocyte. In yellow, we have the beneficial bacteria, Vibrio fischeri. And in blue, we have this other bacteria, Vibrio harvii. And so what I want you to notice is that this hemocyte is leaving a kind of breadcrumb trail of Vibrio fischeri while not releasing the Vibrio harvii in this video. So let's watch it one more time. The blue get bound and gobbled up. The yellow get left behind. And so we want to understand, what is the hemocyte doing to know the difference between the two?
Is the white inside the hemocyte, is that the blue bacteria in phasins?
Yeah, so that's the, both the blue and the pink at the same time. These were all Z-stacks, so for those of us who aren't as familiar with confocal microscopy, it basically takes pictures at different levels of the cell. It kind of takes like a 3D picture, so I can travel through the cell to see what's going on. And what you're seeing in this video is everything squished together so that it's easy to kind of look at like this. But if there's any question about who's inside or outside, I can go through that picture and see what's going on in the middle of the cell. I can also look at each channel individually, but that's a whole other story. Okay, so the first set of experiments I did were with Vibrio fischeri and Vibrio harvii, because we knew that these were the two with the most different hemocyte behavior reactions in the binding assays from the previous work. And I found that the hemocytes released more Vibrio fischeri than Vibrio harvii. And so that's on the left here. Left, yeah. And so then I wanted to add both of the bacteria with the hemocytes at the same time, because I wanted to see if Vibrio fischeri in some way was just broadly communicating to the immune cell to just generally not bind. And what I found was that the presence of Vibrio fischeri did not influence the binding and release of Vibrio harvii. It wasn't Uncle Joe. Okay, that's all I was wondering. All right, so the next thing I did was look at living and dead Vibrio fischeri and different ways of killing them. So over here on the left, we're looking at Vibrio fischeri living. We're looking at polystyrene beads here. I wanted something to give the hemocytes that would have no surface molecules in common with a bacterium that was about the same size as a bacterium. And then I was looking at heat-killed bacteria and then UV-killed bacteria. And what I found was that only about 2% of the beads were released. Meanwhile, the heat-killed were released significantly less, but the UV-killed were not statistically significantly different from either the heat-killed or the living. And so we tried to think about why that may be. We know that UV killing mangles up the DNA inside the cell. And so we think that the surface molecules on these cells are not as mangled as the heat-killed. Because the heat-killed, any proteins present, you might get denaturing of the proteins on the surfaces, and that may account for the differences there. We could also talk about why else that may be at the committee session after. So I next wanted to look at Vibrio fischeri that are symbiotic, but with different hosts. So we have our Hawaiian bobtail squid here. We've got Euprymna tasmanica, they live in Australia. Euprymna morsii lives in Japan. And then we have this pinecone fish, which is quite different. And so I took 4 symbiotic strains and compared the binding and release specifically of Euprymna scolopi's hemocytes, so the Hawaiian hemocytes. And I found that there was no difference between the bind and release of Euprymna scolopi's symbiont between the other squid, but that there was a difference between the squid symbiont and the fish symbiont. So we know that over time, bacteria and their hosts are sort of evolving together. And so maybe there's too much space in evolutionary time between the fish and the squid for it to be recognizable as the same thing. So strain is important in this symbiosis. So next I wanted to look at the surface structures of the hemocyte. And so all of these 4 strains all have modifications in their lipopolysaccharide. So that's like the cell wall component of these bacteria. And they were all statistically significantly different. So we think that lipopolysaccharide is important in the recognition here. And just in case you might think that I succeed a lot, look at all these things that didn't work. We've got a biofilm mutant here, one of which has no biofilm whatsoever, the other of which overproduces biofilm. Neither were different at all. We had a motility mutant, 2 different types of motility mutants that can't swim. We had a light production mutant. We know that this light production mutant can successfully colonize the squid. It can get into that light organ, but it doesn't stick around. It gets eliminated, and we don't know how. So we thought that the hemocytes might be involved there, but it seems in this assay that they're not. And then also a type 6 secretion system mutant. The type 6 secretion system effectively is a needle that inserts effectors, proteins, molecules, into other cells, including eukaryotic cells. So I thought it was possible that they may in some way interact with the immune system, but turns out in this assay to not be affected. I also looked at outer membrane protein U. This is a porin, so it allows for the diffusion of molecules across the cell membrane. And so we had a mutant that didn't have that porin, and it was released significantly less. But interestingly, the presence of the wild-type Vibrio fischeri rescued that mutant. All right, next we have a pretty cool result here. We had squid that I raised in the presence of Vibrio fischeri, and then squid that I raised without the bacteria. And what we found was that, just from previous experiments, that the wild-caught squid, of course, released more Vibrio fischeri than Vibrio harvii. The squid raised with the bacteria also did the same thing, released more of the symbiotic bacteria, but the squid that were raised without the bacteria showed no difference between the symbiotic bacteria and the other bacteria, which really corroborates and brings home the point that you need to have Vibrio fischeri present in the light organ to have the immune system to develop this tolerance to the symbiotic bacteria. So to wrap up this part of the story, hemocytes are able to differentially bind and release different types of bacteria, particularly symbiotic bacteria and others. We also think that the modifications in surface structures of Vibrio fischeri are important in avoiding phagocytosis in the symbiosis, and that surface structures like LPS and OMP-U are important for hemocyte recognition. We think that strain is important in this symbiosis, as we've seen in many other symbioses, and that wild-caught and symbiotically raised squid release more of the symbiont, whereas aposymbiotic squid, so those without the bacteria, do not. So here's our model for understanding how we think these interactions are happening. We have what's called pattern recognition receptors on the surface of the hemocyte, And then microbe-associated molecular patterns, different molecules on the surface of the bacterium that are interacting with each other. And so that may allow the hemocyte to know what it's interacting with. And additionally, we think that OMPU and LPS are very important. And during development, there may be other things that are important here. Maybe secreted factors like outer membrane vesicles are important for developing this tolerance. And so there are a number of future directions here, one of which I'd like to see someone sequence the membrane proteins in the hemocyte and compare those proteins between the symbiotically raised and aposymbiotically raised squid, because we know that they treat the bacteria differently. So is there something different on the surface that allows for that to happen? I'd also like to see some pattern recognition receptors knocked out using CRISPR. We're getting CRISPR up and running at the MBL on our squid, so we'd like to play with that. I'd also like to see when this tolerance first develops, and also look to see at the specificity of strains. So we have multiple strains from the Euprymna scalopes kind of group of strains, and if you raise a squid with just one of them, does it know the difference between that one and another squid? That would be interesting, which is different from what I've already shown. So I have a lot of people to thank during my time here, my PhD, my whole lab, starting from the get-go with Andrew and Tyler and Allison and Andrea and everyone. It's just been a great ride. And also every undergrad who has ever helped with the Squid Room. We've got one of them here today. She's MVP, because then we don't have to be in there all day, every day, just most of the day, most days. I'd like to thank Chris O'Connell and Carol Norris for helping with microscopy over the years. Tim Moore for helping with statistics this past month. Everyone in my committee, my mock committee, for helping make this talk more digestible. My family, my best friend Jenkins, and my roommate Nick, and everyone in the MCB crew who has ever helped me. Thank you to Tony, who's talked to me about academia and Squid and science and everything I would ever need over coffee or beers or whatever. And then Jason, who every time I couldn't lift a tank, or I couldn't lift it by myself. He is a computer person, so he's like beep booping up on the second floor, and I would just be like, Jason, come help. So he's helped in the Squid Room like almost as much as some of our undergrads. And anyone who's ever come to one of our stupid theme parties, appreciate it. And of course, Spencer, who's been with me and really allowed me to do a lot of goofy stuff and hasn't guilted me for that over the years. He, of course, went with us to Japan, and he really just maintains this sense of wonder in nature that I really sincerely appreciate. It's just inspirational. So, and then of course my lab mate Andrea, she started when I started. She graduated 2 weeks ago, and now I'm finishing up. So we were in together, out together, and she's gone squidding with me. She's gone all over the place for conferences, and grad school would not have been the same without her. And lastly, my best friend Jenkins, who's phenomenal. He also went to Japan collecting squid with us and like ran through fish markets and got his hands dirty with a lot of squid guts, and boy, do I appreciate him. And also, he's helped me with Skype a Scientist a lot, and I really appreciate it. So now I'm going to take any questions, truly any questions. If you have a question, you may ask it. Don't worry about stupid questions. There's no such thing as a stupid question. Great. Tony.
Your model for the first half of the talk, correct me if I'm wrong, but you have this pore formation of the ANG, bacteria land, there's invagination, tubules are formed, and then more pores come, you get more landing, and as the animal develops, you kind of get invagination. So, to your hypothesis about the root microbia, then, do you see when you section through the ANG that the root microbia are in the deeper tubules? So if you were to take that organ, is that?
We haven't done that, but that's a really great experiment. One thing that I've been playing with doing, if I can squeeze it in before I leave, is, or maybe Derek might do this too, is fish on the first, like in early development of those pads. We want to do whole mount fish to see if we get Verruca microbia there first. And then, but the sectioning through might also be cool.
Thinking about the sequencing data there, it suggests that there's early colonization by Verruca microbia and then
Yeah.
bacteria.
Yeah.
So just holding that organ, right, you should be able to section through, unless if you get rotation of tubules for some reason.
It's possible, but that would be a fun thing to, as a start, to see what's going on in there. Yeah, thank you.
Kind
Mhm.
right? Did you
Yeah.
ever grind up or fractionate ANGs?
We did. All the squid died. I don't know what to tell you. All the squid died. Yeah, that was before I had like made this whole big Franken system of flow through machinery. So we could try that again. But when we were doing it, we're doing water changes every day, and there wasn't quite enough flow. We got this nasty biofilm forming like overnight. It was pretty nasty, and the squid just couldn't hack it, unfortunately. Squid are pretty delicate animals, so I don't know. Didn't work out. Andrew?
So, most important question. Of all the squid in the world that you've looked at, how conserved is Veracruz? Is it really unique to...
It's not very conserved. Verrucomicrobia is not present in all of them. So another question that we would have then is why would Verrucomicrobia be so important in Euprymna scalopes when it's not even present in some of these other squid? And we don't have a good answer for that, but maybe it could be that there's another bacterium that's important for the cuttlefish, for the Doryteuthis. We don't know. I'm ready.
Can I ask a follow-up to the first question though in between?
Mhm.
So, but have you been able to look at the developmental stages that might have improved
Right,
in
no.
other species?
We haven't. We do have, I've looked at the development of parva, which was the little blue squid in the bottom right corner that had the orange ANG. So those animals, we've looked at smaller animals, and the development looks the same in terms of the morphology, but we haven't done sequencing on smaller things yet. Once we're out in the field and have more time to play around, I think that'll be something we'd love to look at. And we also have a group of people at the MBL that are raising these squid up. So we can sort of play with that as well, hopefully next summer. Second question.
So I'm wondering about all the squid moved to Japan. Like, do any of them like overlap geographically? Because I'm wondering, like, is geography a factor in like what bacteria they pull in?
Right.
Or is it more
So that's what we'd like to know. So we are working on getting, so Sepia-teuthis are those ones that are yay big with the kind of oval shaped fin. And so we have a collaborator in Africa that is going to collect them. We've already collected some from Japan, and then we're hoping to go down to Florida to grab some of their different species but same type of squid and take a look at whether the community is stable over very different geographical areas. And then the reason we went to Japan was because there's a number of different cephalopod types all in the same snorkeling place. Like we went out and saw a cuttlefish and a parva and an octopus, like all within the span of a half hour. So we've got plenty of cephalopods there to work with. Australia would be another great place because you have the Sepia-teuthis, you've got the cuttlefish, you've got other squid, you've got a lot to play with in the same body of water. And so we need more data to really strongly say it's geography or it's relatedness, but so far, tentatively, we're saying, I think it's relatedness. Yeah. Tony.
Did you test, say, catch and release assay or anything associated with that with any of these strains isolated from the AMG to look for recognition and compare that to strain?
Not yet. Andrea worked on trying to get a fluorescent strain of one of the ANG bacteria, ANG1, but they weren't very bright, and so that's what she's actually doing her postdoc on. And so I think NIDI is going to be continuing experiments with the ANG bacteria because we are curious of the differences between, for example, male and female squid in that response. Katie Kyle? So some people have cultured some other Verrucomicrobia. They're very slow growing, and other than that, I don't know, but Andrea recently got a genome of Verrucomicrobia, and so we've been looking into that to try to figure out what sugars do these guys need. We're gonna try mucins. They just might need different stuff than we're giving them, and so we're gonna be playing with that.
When the ANG forms,
Uh-huh.
is it always the same crypts in the same place, or are they just growing? Like in your pictures, where you can see bacteria are in different crypts.
Uh-huh.
Is it always that same pattern, or is it just separated?
In the light organ crypts, it's very much, everybody pretty much looks the same. There's always 3 on both sides. There's always one that's a little smaller than the other 2. And so in the ANG, we don't have that much information there yet. In adults, there's a decent amount of variation, even in size of ANGs, and the ones that we raise are quite different from each other. Like, I think that might be my first slide. So here's some ANGs showing like kind of the variation that you see in the raised ones. And we kind of think that the longer the sand has been in our lab, the worse it is at making a robust looking ANG. Like this thing's pretty measly looking. Like it has different colors in there, and clearly there are tubules that are developed, but it's nowhere near the size of a wild ANG. Austin.
How homogeneous are the bacterial communities in marine systems, like the sand versus when they come out?
Allison, my former lab mate, did a whole transect of many in one beach, but I don't remember how stable they were. Do you? I want to say they were stable. They were stable. Cool. Yeah. Yeah. But like, how different would it be on the other side of the island? I don't know. There are certainly different bacterial communities in different, like, way different, like, continents apart. But yeah, I don't know. I don't know what differences you would see in like Hawaiian seawater versus Japanese seawater. I don't have that off the top of my head, unfortunately. Aoife. Totally. Yeah, we've got some very odd-looking stunted ANGs. So back when I was first starting, I just wanted to see if I could get the squid to live. I wasn't concerned with what bacteria I was giving them. I was just like, let's just get healthy, happy adults. And in that system, I was raising them on sterilized sand, sterilized like filtered water, in these little tubs in a little closet. And then once they were big enough that I had faith that they could survive in the bigger system, I moved them over. But adult females were laying eggs in those tanks. They were living and dying in those tanks. So we think that there's a decent amount of bacteria from the ANG in that system. And so I think I was putting them in that system in the late stages of their recruitment. So some bacteria did get into those ANGs, but they were very strange looking and very small. So you could get a weird looking ANG, but that was a pretty lab kind of situation. Like I've never seen that in the wild. You don't, it's not like a switch at a very specific time point. There's a pretty long open recruitment call, like kind of rolling admissions for symbiosis. But we haven't really pinned down when the last second that you can get a bacterium in there would be. JD. The light organ, yes. The ANG, we think the bacteria only get cycled through when the female's laying eggs. We think the rest of the time it's just hanging out. So my former lab mate, Allison, looked at the stability of the symbiosis in laboratory culture to see if it changed at all once coming into the lab in cleaner conditions, and it's pretty stable. Yeah, so once you have the bacteria in there, you've got them for life unless something strange happens, we think. Aunt Regina? Around 700.
How did you determine which species you wanted to study?
Great question. So of all the squid, only some of them have ANGs. So you have those like deep sea squid, the colossal squid, the giant squid, no ANGs there. Let's not even worry about how you would get one of those, but they don't have ANGs. All right, and then some market squid have them, some don't. So we're just looking, well, when we were trying to decide what squid we're trying to get, we were trying to find a lot of ANG having squid in one area. So there may have been other squid there were other squid in the area when we went to Japan, but they didn't have ANGs because they were from a different group. So of the squid, there's squid, there's cuttlefish, there's bobtail squid. The bobtail squid all have it, the cuttlefish all have it. The squid, which is the hugest group, there's something called a myopsid squid, and they have it, and then a gopsid squid, and those are most of the weird deep sea squid, and many other squid as well. They don't have it, so we were basically just going on who can we get our paws on that would have an ANG. Let's sequence those. Katie. A lot of them just carry them with them. So you'll see, if you've ever seen a picture of these squid that have this like huge mass of eggs in between their arms, just swimming along with a big mass, that's one way of doing it. And then also Humboldt squid, which are those big, nasty human-sized squid off the coast of California, they have these huge gelatinous, like car-sized mats. And I don't think they have bacteria in them, or at least not from an ANG. I don't know if they're producing like antimicrobial peptides from the host. Not the host in this case, it's just the squid. Not sure. But there are other animals that don't have it that have these like tough outer casings that make stuff hard to get in. I'm gonna not, Tony, you've asked too many questions. Corey?
Have you treated your squid with antibiotics and disrupted that microbiome to see what happens to these different organs? Like, do you get a collapse of the ANG, or
Yeah.
do you reset how they interact with Vibrio fischeri?
So we have done a number of antibiotic experiments. One with eggs. This was, again, Allison. She treated the eggs with antibiotics and then treated them with fungus to see what would happen. And the ones that didn't get antibiotics were, for the most part, fine. The ones with antibiotics all got totally fungal infected. So that was evidence that the bacteria really are doing something. The other thing we've done is looked at a lot of curing experiments with the light organ stuff. And so that first slide that I showed on the binding from Spencer's stuff, he cured the squid and then over the course of 5 days monitored binding and found that more bind, the squid hemocytes will bind more of the beneficial bacteria when cured. So that indicates that really you constantly need that Vibrio fischeri in the light organ to keep the immune system operating normally. In terms of the ANG, I think there was an experiment done where we cured a squid and then looked at the ANG, and you don't get rid of all of the bacteria in there. I've also, but I don't remember who survived and how healthy the squid were later. Another thing that we've done, when I raised the squid without ANGs, I mated them just to see, can they even make an egg without an ANG? And they do, and those eggs do hatch. But keep in mind, we're raising these little eggs in a pretty clean system. I mean, it's in TLS, but it's still pretty clean. So TLS is the next building over, it's not. And they were all right, you know, but I think if we treat those with fungus, that should be an experiment that the next cohort does. Becca.
Why would you harvest dry sand instead of wet sand?
Sure. Yeah. It's a stupid answer, too. So the answer is, this guy, Eric, told me that the squid do well on this really fine sand. And it's true, they do great. If you don't care about the bacterial community in the squid, they do great because it's really, really fine, and these itsy-bitsy little squid can easily bury in this fine, sugary sand. That's why I collected it. I collected it because I wanted my hemocyte experiment squid to be happy when I was raising them, because at the time, I didn't care as much about the ANG development. I hadn't started that project yet. So that's why it was around. That's why I did that.
So all the time you've been raising, okay. So if you guys can't harvest that Vibrio microbiota,
Uh-huh. Mm-hmm.
how have you guys been growing squid beforehand if you guys didn't have the wet sand from which it comes from?
Well, we were raising them for hemocyte experiments, so it wasn't, we weren't thinking about it, really. It may be important, but we weren't thinking about it. And then, really, we've only been raising squid hardcore since I got here. Andrew did one cohort that did all right, but so, yeah, we haven't spent much time thinking about it until now. Yeah.
Do you think so the initial inoculum comes from like when you bring back squid from Hawaii, something like that?
Yeah, so when squid die, we think that the ANG gets really mushy very quickly. It decomposes very quickly. So that bacteria could be getting into the sand. But we haven't tried to raise squid on the sand from the tanks because we would just rather it be more controlled than the happenstance in those tanks. Do you have a question, Lori?
Yes. So do you think that the hemocytes will respond differently in the catch and release assay if you use hemocytes at different stages of development?
We do. Yeah. And I have some preliminary data for that, but I hadn't finished it off before graduation.
Because the B. fischeri colonize earlier than the B. harvii and you know, allow the hemocytes to like remember them.
Right. So the Vibrio harvii is never associated. That's just like a bacterium that lives out in the seawater. So it's just like a control of not symbiotic. And so we think, well, one of the things that I think a future direction should handle is at what point in the squid's development with the Vibrio fischeri does that bind and release tolerance start? because we have data from little hatchlings, and they don't really do very well at telling the difference, and so we don't know when that starts. Yeah, but we'd like to know. Corey? Oh, great, okay. Melanie?
So what's the typical lifespan of the squid, and how did that differ from your? I know that you said they could live the full life in the lab. But how much did that differ?
That's so the ones that we raise in the lab live about 6-7 months. We previously thought they lived like 9 months, and maybe they do longer in the wild, but they also have things eating them in the wild. So on average, I think they probably live longer in the lab. Kathy?
This is a real civilian question, but how do they travel? Like, how do you get them back from Japan here without?
Yeah, so from Hawaii, we put them, you ever go to like the carnival and there's like a little fish in a bag? That, in a cooler, and then we just ship the cooler back on the airplane. It's in the cargo hold while we're in the regular hold, and that's how that happens. In Japan, it's hard. So what we did is we sacrificed the squid in Japan, and then we put them in like a DNA preservative, and then processed them when we got back here. Because getting an animal through customs fast enough for it to survive is hard, and we didn't want to risk it. The heat and cold is really hard, and when we're shipping them back from Hawaii, that's also really hard. Sometimes if they miss a flight, they're screwed, yeah. Yeah, it's really sad.
Any more questions? All right, let's thank Sarah.
Oh, stop. Stop it. You're embarrassing me. Okay.
Now, Sarah will meet with her committee, and then we'll meet later on up on the floor.
Yeah. So, family, where's Rose? All right, you're going to follow Rose from here.